Okay, so we are now recording. And well, it's 3:33, so I'm gonna get the show on the road. So welcome everyone to our November Chairs Lecture of 2020. And I have the great pleasure of introducing today's speaker, Rich Palmer. Rich will be no stranger to many of us. Rich is a professor emeritus in the Department of Biological Sciences. And I can give you a few highlights of his background. I'm not going to read his whole CV because there's a lot of stuff in it. But Rich started here. Well, let's even go back. Rich did his BSc at the State University of New York at Stony Brook, PhD at University of Washington in Seattle, and then joined the Department of Biological Sciences. Well, in fact, it was the Department of Zoology, right, at the time, in 1980. Rich rose through the ranks here, as well as having stints at Banfield as director at Banfield for a number of years, to become a full professor, I guess, in 1991, and you retired in 2017. So I reckon that's about 37 years of service to the Department of Biological Sciences or Department of Zoology.
40 as of the end of December.
Right. Yeah. Okay. So that's quite a milestone, really. 40. Wow.
It's a long stint.
Yeah. Well, Rich has had a stellar career. There are many highlights, many awards, and I'll just point to a couple. He's a Kaplan Award winner, which is the highest research honor the University of Alberta bestows on one of our academic staff. He's also a fellow of the Royal Society of Canada, and there's a long list of honors. I'm just going to share one little story about Rich, and in fact, it's a story about Rich that goes back to before I even met Rich. When I was, my first academic position was at the University of Sheffield, and I started there in 2000 in England. And I remember when I left Sheffield in 2004, and I had this position, this offer to come to the University of Alberta. I remember having a chat with people in the coffee room, and one of my colleagues said to me, "Oh, that's fantastic, University of Alberta. I know Rich Palmer is there." And he's so, Rich, your reputation really does precede yourself, because you're the name that really came up. And the reason it came up is because we'd actually been doing journal clubs at the University of Sheffield. And we were doing journal clubs, and particularly on fluctuating asymmetry, and actually anything that had Anders Moller's name on it, we were doing journal clubs on. And Rich, if you know anything about Rich's work, he's done some really important work about fluctuating asymmetry and other forms of symmetry throughout his career. But I remember doing a journal club on a paper called, I think it was Quasi-Replication in the Contract of Error, which was really about meta-analysis and selective reporting. And that was a really important paper for me, because I remember using that paper in the first meta-analysis I wrote. But the fluctuating asymmetry stuff was really what people were buzzing about at Sheffield, and the discussions with Anders Moller that you were involved with, which kind of put University of Alberta on the map in a little bit of a way, all the way over there in the UK. So I remember him saying to me, "Well, that's great that you're going back home to Canada. I know you've always had a yearning to go there, but you get to work with Rich Palmer, and that's just amazing." So Rich, your reputation precedes yourself. And this was some time ago now. With that, I'm going to hand over to Rich to talk about one of his latest pieces of work. And with that, Rich, the screen and the floor is yours.
Okay, so I guess thanks for those kind words, Dave. I very much appreciate it. I just can't leave fluctuating asymmetry behind me, I guess. And thanks to all of you for taking time out of your busy day to listen, particularly given the extraordinary distractions going on south of the border. I only wish I could be doing this in person instead of from such a distance. So as I was telling Dave a few minutes ago, you'll no doubt be surprised to hear that this is my very first Zoom lecture because I'm in a phased out retirement. I don't teach winter term, so I haven't had the privilege of doing Zoom lectures. However, in about 2 weeks, I get to do the last course of my phased out retirement, and it will be via Zoom. So I must say, I'm really looking forward to it. And just for the record, I'd like to confirm that I am wearing pants, as I've heard that Zoom lectures are pants optional, but I decided not to take advantage of that facility. So let's see if we can get this started. So Dave, can you confirm that that's behaving as it should? Okay, great. So what I'd like to do is introduce you to three remarkable organisms that are remarkable because, first of all, they can make pretty large sounds, surprisingly loud sounds, but they can also, they are also surprisingly small for creatures that make such small sounds. And so I'll be talking about an example of work that I've done on shrimp that snap with specialized claws. Another example with an amphipod is a very small crustacean that also snaps using a specialized claw. And then the last example I'll talk about are some worms that remarkably are able to produce these very loud sounds just using a muscular tube. So I'm actually less interested in the sound production of these systems than in the biomechanics underlying how they produce these sounds. And I'm also particularly interested in the evolutionary sequence of steps that gave rise to this remarkable ability. So for example, how do you get shrimp that can make these spectacularly loud sounds from ancestral shrimp that just had very tiny little pinching claws? And then if there's time at the end, we'll see, I'll spend a few minutes reflecting on a 40-year career here at U of A in the hopes that there might be some lessons there of use to some of you. So, oh, well, that's interesting. My PowerPoint is not behaving. Let's try that again. There we go. So the first is sort of an audience participation test. How many of you recognize this sound? You can wave at me if you recognize this sound. This one's a pretty easy one. It's a humpback whale. Here's another one. Again, wave if you recognize that sound. It's probably a cetacean. And you're right, it actually is a cetacean. It's a harbor porpoise. And then what about this one? This is what you hear if you put a hydrophone under the water in Grappler Inlet, just around the corner from the Banfield Marine Sciences Center in Banfield. And I'll talk a little bit about this towards the end of the talk because it's an area of ongoing work. So just to start with, how loud are these snaps? So as I said, we're talking about three examples, snapping shrimp, snapping amphipods, and snapping worms. And we do have data on sound levels in snapping shrimp and snapping worms, and they're seriously loud. So here's some other sound levels from other systems, both marine animals, but also for reference, the threshold of human pain. And you can see that the sound intensities, the decibels that snapping shrimp make and snapping amphipods make are well above the threshold for human pain and up in the range of rifle shots and also in the range of some very large mammals. And yet these creatures are all quite small. So what these three systems share in common is a way to move parts of their body extremely fast. And that turns out to be much trickier than you might think. All three of them are able to produce cavitation bubbles, and a cavitation bubble is what looks like a bubble, but it's actually a vacuum. And after creation, it just that vacuum bubble collapses under its own ambient water pressure, and it's the sides of the bubble coming together that make the loud snap. So all three of these systems, we think, make loud sounds via cavitation bubbles. And in order to do that, they have to move super fast. In fact, they have to move faster than is possible just by simple muscle contraction. So these and other animals all use some system of springs and latches. And this is the best analogy, which I've stolen from Sheila Patek, about how sort of a spring latch system works. Basically, you can use muscle energy to contract slowly and store a tremendous amount of energy, and then suddenly release that stored energy as you would release an arrow out of a bow. And the arrow will travel much farther because that energy is released much more quickly than if you simply took the arrow and tried to throw it. So all of these three systems have all of these three creatures have some system of springs and latches, but as you'll see, they're quite different in the different animals. So let's start with the first one. How does that sound come through, Dave? Is that good? So this is Synalpheus. It's the largest species in a genus of snapping shrimp. And there are other larger species of Alpheus, another genus in the family Alpheidae, but these guys can make really quite remarkable sounds with these huge claws. And that's been known for decades. But it's only about 20 years ago when someone finally figured out how they make these sounds. This is what the claw of a snapping shrimp looks like, and there's some really unusual features of it. But originally, most zoologists thought that the snap was created by the impact of this finger closing, the movable finger closing onto the fixed finger. But in 2000, finally, the technology had advanced enough that it was possible to film these animals at super high speed. And this is what actually happens. So these are videos, actually fairly low tech given what we have now, but these are video sequences filmed at over 40,000 frames a second. So this is the side view of a claw, and this is the top view. And you can see they close extremely rapidly, and there's this what appears to be a bubble that emerges, which is the cavitation bubble. And you can even see as the bubble collapses, the sides close, and then they impact each other, and it's the impact of the sides of that bubble that creates these very large sounds. So the reason they're able to generate these cavitation bubbles has to do with the physics of water and high-speed movements. In an Alpheus claw, the movable finger has a big tooth that inserts into a cavity on the fixed finger. And if you all remember first-year physics, water is incompressible. And so as the claw is closing and this finger goes into this tooth goes into this cavity, all the water that's in that cavity has to leave because it can't be compressed. And it's guided out through a narrow slot here in the front to move so fast that the adjacent water cannot keep up with it. And it literally tears it apart to make these cavitation bubbles. To give you an even bigger impression or better impression of just how powerful the energy is released from these collapsing cavitation bubbles, have a look at this. So this is a high-speed video of a claw closing, and I think this will repeat for you. Watch right at the tip after it closes. You'll actually see a flash of light. So there's so much energy released when these cavitation bubbles collapse that there's a momentary release of light at extremely high temperatures. So that's the most familiar group of shrimp that snap, but there's another group of shrimp that snap. And this is a wonderful little example of them. And it's another family called the Paleomonidae. And this animal is only about 2 centimeters long. It's not a very big shrimp. And what you notice a couple things, a couple of interesting differences between this shrimp and the snapping shrimp. One, this animal has 2 claws that snap instead of 1. And even secondly, they have a tooth cavity system just like in the Alpheus. So here's the plunger, here's the cavity. As the claw closes, water is jetted. But look at where the tooth and the cavity are on this claw compared to this one. They're reversed. Here the tooth is on the movable finger, here it's on the fixed finger. Well, these are two, and they're also on a different limb of the body. So P1 is the first thoracic walking legs. P2 is the second pair. So they're on different legs and they've got a different arrangement of tooth and cavities on them. So both of those suggest independent evolutionary origins. And then with a postdoc of mine, we tried to reconstruct that history. So with my late postdoc Tominari Kaji with Arthur Anker and with a German colleague, Christian Werkner, over about 2 years, we collected a massive amount of data and put it together to try to reconstruct the history of snapping in these 2 different families of shrimp. This involved trips doing fieldwork in Panama, the Solomon Islands, Great Barrier Reef, some work in Bamfield, and then in Rostock, Germany. Most of the work involved doing micro CT or confocal images of claws from a huge diversity of shrimp. So over 100 different species of shrimp from 19 different families. And then Tominari Kaji also did a bunch of high-speed filming of different claws, as well as some kinematic experiments with enlarged 3D printed models of claws. And that allowed us to reconstruct, I think, quite convincingly, the evolutionary history of snapping within the Caridia. It's the largest group of shrimp. These are the so-called the true shrimp or the Caridian shrimp. They include a huge diversity of shrimp, a few thousand species spread across more than 28 different families. And here's just a sampling of them, some amphipods, but also some other more colorful ones. And here are the 2 families in which we know that we have species that snap, the Palaemonidae and the Alphiidae, and they're not even sister taxa, so that further reinforces the idea that these are independently involved. So the first step was to look within one of these families. So take the Alphiidae here and ask, what can we say about claw form and function across species within this family Alphiidae? And this is where Tominari, the wizard, was able to figure out some crucial things about joints in claws. So here's an example of a claw from Salmoneus. And what Tominari would do is he would image these claws in 2 different arrangements, one with the claw fully closed and one with the claw fully open, looking very closely at the base of the claw to try to figure out how that movable finger moved. And this is what he found. What he found is that he found a joint that people hadn't realized existed. Before this work, almost everyone believed that crustacean limb joints, certainly crab claws and shrimp claws, the movable fingers simply pivoted around a single axis. But what Tominari found is that, at least in this particular genus, the base of the movable finger slides over this ridge. So it's a very different kind of movement than simply pivoting around a single point. So here's a little cartoon of what that means. So if you look at a reference point on the claw, you can see it isn't just rotating around a central axis, it's actually sliding around this ridge. And as you'll see a little bit later, that's extremely important. It turns out you can recognize a joint and whether it's a slip joint or not just by looking very closely at the base of the movable finger, the dactyl. If it's curved, it just rotates around a single point. If it's really flattened, you know that it's sliding over this ridge. And then we see some in between where they're slightly curved. So it's possible just to recognize this morphologically. So the next step was to look at a bunch of different species in the Alphiidae. So we're back in the family of shrimp that include the familiar snapping shrimp. And with an exception of one clade, and if anyone is interested, I can talk about that later, all of the Alphiids that he looked at had this slip joint. So here's an example of the variety of different claws filmed open and closed, where Tominari could show that the finger was sliding over this ridge. So what that allowed us to conclude is that most of the alphias, or at least all the ones we looked at, except some of the derived ones, had this slip joint. But what about this second family in which we observe snapping? So here's four of the examples of species that we looked at from within the Paleomonody. And same thing, all four of these, when imaged closed and open, we could see that these joints at the base of the movable finger were slip joints. So both within the Paleomonody and within the Alphiidae, almost all the shrimp that we looked at appeared to have slip joints. So that then begs the next obvious question: what about more basally in the Caridia tree? And so we sampled a bunch of different taxa in these more basal caridians. Here's just an example from Pandalidae. And lo and behold, even some of these more basal ones have a slip joint. And in fact, yeah, so again, there's a close-up showing this movement where you can see this reference point actually sliding around the edge of that ridge. Here again, all of at least these intermediate taxa, let's say, were among them, many of them show the slip joint as well. So this leads to the inescapable conclusion that, in fact, slip joints evolved long before the ability to snap, and in fact, very early on in the radiation of this huge family of shrimp. What about at the very bottom part of the tree? So here's a taxon from the very basal part of the tree. And it exhibits the more classical pivot joint where there's a single axis and the claw simply the movable finger simply pivots around this central axis of the finger. And here's an example of a schematic of what that simple pivot movement looks like. So the slip joint is obviously shared by a lot of different Caridian shrimp, but the ability to snap only evolved in these two groups up here, the Paleomonidae and the Alphiidae. So now the question becomes, how do the snapping slip joints differ from the other slip joints in shrimp that don't snap? And this is where we took advantage of some very cool technology where you can take micro CT images and you can threshold out parts of the image, in this case, the cuticle of the claw. You can send it to some people that can print 3D models and make very large, 50 to 100 times life-size models of these claws to better understand how the movements work. And this turned out to be really critical to making the case for how these slip joints work and how they're able to generate such rapid closure and snapping. So here's one of these 3D models. So that's about, 25 centimeters long. In reality, the claw is only 2 to 3 millimeters long. And watch what happens. This line is actually attached to the base of the movable finger, where in a normal claw, if you pulled on it, it would pull the claw closed. So these are videos that Tom and Harry Kaji took. Look how, as the angle changes and you pull it, nothing happens until the angle is quite low. You get quite down towards the bottom of this very large closer muscle. So let's look at that one more time. So it's pulling hard and the claw doesn't move. So any contraction in this region doesn't actually move the dactyl. It's only contraction down here that starts to move it. So what that means is that at least initially, a lot of the muscle in the claw, when it starts to contract, is not moving the tip of the dactyl. It's simply contracting and storing energy. So what happens in a non-snapping slip joint? Here, there's one apodeme that attaches to the closer side of the dactyl and another that attaches to the opener side, and they simply antagonize each other around this sliding joint. And the key difference is that in the snapping slip joint, a lot of the muscle is actually involved in energy storage before closing. And so these muscles can contract, massive amounts of energy can be stored. until the muscles here on the other side of the pivot, so this is the fulcrum, the muscles on the other side of the pivot eventually cause it to slide. We didn't discover until after we had worked out the mechanics of this joint that this joint actually has a name. It's called a torque reversal joint because during movement, the torque changes from pulling clockwise to pulling counterclockwise. So here's what that looks like. So energy storage and cocking when the muscles that are attached to the right of the fulcrum, and then when the muscles to the left of the fulcrum contract, they pull the tip of the dactyl over that ridge, and then it closes. And all that energy is released suddenly. In the non-snapping slip joints, you still have opener muscles and closer muscles. So these are the opener and closer. But in this case, the closer muscle never goes beyond the fulcrum. So any involvement in closing closes the claw. So that's the key difference between a slip joint, which can store energy and then close quickly enough to snap, and a slip joint that is just a simple non-snapping slip joint. So the punchline from all of this in our current biology paper was really one of the origin of novelties or innovations, and it's really several. The first is the evolution of this simple sliding joint as opposed to a pivot joint. Happened very early in the evolutionary history of the Caridia, this very large group of shrimp. And then two independent origins of snapping slip joints, where some of the closer muscles become involved in energy storage and latching. So that's the first example I wanted to talk about, the most familiar example and one about which the most is known. The second example is it's just mind-boggling. It's this amphipod is only about maybe 15, 20 millimeters long as an adult. And watch this movement. So this is a video that doesn't have any sound, but my postdoc, Tominari Kaji, had a colleague who knew about these amphipods, and he said, oh, here, let me send you a video we took. And they sent us this video, and we both looked at this video and instantly realized that these animals have a claw that closes, that clearly is involved in latching, energy storage, and then extremely rapid closure. And there were anecdotal reports in the literature that they make a snap, but it had never been quantified. So I set out with Tominari to go to Duke University Marine Lab to work with Dr. Sheila Paddock, who's very well known in the high-speed movement area, and she has super fast video cameras that can film at almost 500,000 frames a second, some very high response rate hydrophones, and she's also very good at working out the mechanics of these unusual high-speed movements. So here's a rather crappy video of an amphipod just shot with a handheld camera, but it does have some audio. So listen to this carefully. It'll repeat. You'll hear two different sounds. One is a pop that's more like the pops that you've already heard, and then another is something I call a ting, which almost sounds like banging a wine glass with a knife. So listen closely. Oh man, can you hear those? On my earphone isn't good enough to pick them up. There's a pop, and then a very high-pitched ting. It may be better on your speakers than on my earphone. I know my computer speaker's better. Anyhow, they're two very distinct sounds that these animals make. But these are quite remarkable animals. First of all, they're sexually dimorphic. It's only the males that have these really hypertrophied claws, and they only have them on one side. These claws actually get up to be about 25% of the body weight of the males. It's a massive investment in a claw. It's only on one side of the body. Half of them it's on the right, half of them on the left. It's another reason for me to be interested in them. But the dactyl closes so fast that you cannot resolve it with video at simple 30 frames per second. So here's what we were able to see when we finally got these animals set up in front of a really high-speed video camera. So this is 80,000 frames per second filming, playback at 30 frames per second. And there are a couple of things you can see. First of all, as that finger closes, there's a jet of water that shot out from the claw. You can see that sort of it's almost like echoes in the water here. That's probably dimpling of the water surface. But look at what happens to that claw after it snaps. It torques almost 180 degrees. It's an incredibly violent response. And then here's another high-speed video. This is 300,000 frames a second. And you see some of the same thing. There appears to be water jetting from the medial side of the claw, so away from you, out of the field of view here. There's also really violent recoil, about a 90-degree twist. And if it sort of looks like something funny is happening at the end, you see what looks like a little bit of a flash. So let's look at that more closely. As we looked at these more closely and slowed them down, what we found is there appear to be two different ways that cavitation bubbles form. So here's a sequence from one clip where as the dactyl closes, you actually see about 1/10 of a millisecond after it starts to close, a little bubble forms behind it. And then as the claw eventually completely closes, that bubble disappears. And you can see that in this video, although it's a little hard because it happens so fast, but you see most of the flash occurs behind the video, or behind the dactyl rather, as it's closing. So this is a cavitation bubble that is not produced by a jet of water. It's actually produced at the trailing edge of a very rapidly moving structure, much more like cavitation bubbles do in boat propellers. And then here's the other one. So this is where you can see what looks like a brief flash here on the inside of the claw. So there's another type of cavitation. Here you can see that the bubble first appears about 1/10 of a millisecond after the claw closes. And then it enlarges medially. So there's clearly an enlargement of this bubble towards the midline of the body. That bubble collapses, but then here you can see traces of a recoil. So the bubble collapses, it bounces, it pops apart again, and it closes a second time, all within about 1/10 of a millisecond. So how do snapping amphipods and snapping shrimp compare? The snapping amphipods are much faster than snapping shrimp, so they can close in about 1/10 of a millisecond compared to 6/10 in the snapping shrimp. In this type 2 cavitation that we observed, the water jetting is actually towards the medial side of the claw instead of from the tip of the claw like we see in Alpheus. And then the bubble collapses much more quickly in 3/100ths of a second in snapping amphipods than in snapping shrimp. So Sheila Patek and her students, so lead author is Sarah Longo, in a paper we have coming out in Current Biology in the next couple of weeks, I think. It's been accepted as just in the final stages of production. She measured both, quantified much more precisely acceleration rates. And what she found is that the accelerations of these claws as they close is among the highest known in animals. So what's summarized here are acceleration as a function of body mass. And there, some of the animals that can achieve these very high accelerations are nematodes, insects, frog jumps, mantis shrimp mandibles, and our appendage strikes. There are some other much higher ones here, but it turns out all of these higher accelerations are actually in mandibles in insects closing in air. So these snapping amphipods are right up there among the very fastest accelerations in animals in water. And here's another way of looking at it. These are just the angular velocities that the tip of the claw can achieve. Here's the angular velocities in amphipods. It's over 1,000,000 degrees per second. It's unbelievably fast, and that's more than 5 times as fast, almost 6 times as fast as snapping shrimp and stomatopods, which were the other fastest moving animals in water. Here's some animals in air. So trapjaw ants have a similar structure or a similar arrangement where 2 structures come together. And even they're only about half as fast as snapping amphipods, whereas in termite mandibles, a couple of, and Dracula ants, they snap, they cheat. They actually push their mandibles together and the mandibles stick and then they release like that. So it's a very different kind of a movement. But anyhow, these amphipods now hold at least the water speed record for angular velocity. The claws, if you look at them, have some really interesting characteristics. They've got these distinctive teeth, the function of which we're not sure of, but they've got this propeller-shaped dactyl that fits very tightly into a groove. We think that's really critical for the production of these water jets and eventually the cavitation bubbles. But the question that remained is how do they achieve these incredibly high speed movements? How do they latch? It's pretty clear something is latching, energy is being stored, and then they release suddenly. And here's our guess. It looks like the latching system is this very strange 2-part tendon. So this is the movable finger of the claw. This is the fixed finger of the claw down here. And if you look at a blow up of this tendon, so these are all the muscles that attach to this tendon. And when they pull, they should close the claw. But this articulated tendon has a second piece with a tooth on it. And what appears to happen is this tooth gets lodged on a ridge on the palm of the claw. So when the muscles originally contract, it can't close. The movable finger is stuck until this slides off the ridge, and then all that stored energy is released and the dactyl can close very quickly. So it's and so far as we know, no one's ever noticed this type of latching system at least in a crustacean joint before. And it's something that we're currently working on, trying to see what this looks like in other amphipod groups. So I pointed out that there were 2 different sounds that these animals make, this pop and this ping sound. And it turns out we know a little bit about the popping sound. So if you look at these videos here, you can see the dactyls are just twitching a little bit. Do you see that movement? It's very subtle. So what the student who did this did is he stopped these high-speed videos precisely at the point that he heard this popping sound. So this tells us pretty clearly that the popping sound is actually associated with unlatching. And we suspect that really glass-like tinging sound is associated with or derived from cavitation, but that's yet to be confirmed. So the third example is it's too weird for words. I mean, who would imagine that worms could create loud popping sounds? So get a load of this. It's completely crazy. These are worms that live inside glass sponges in fairly deep water. They appear to defend territories inside these glass sponges using this really nasty type of fighting we call mouth fighting. And associated with these strikes are these really loud sounds. So that's quite remarkable that they can produce these loud sounds, and it really begs the question about how could something with a simple muscular tube actually create such loud sounds. Well, there's a little bit of a hint in this video. So in this video, you'll see 2 worms, and I want you to look really closely at the posterior end of the pharynx in both of them. This one will go first. So they're getting ready to strike, and you see this contracts, it gets narrower and narrower, and then it opens explosively. And then the same thing happens here. Actually, that's even slowed down because the pharynx was obstructed. So it's pretty clear that the rapid expansion is just associated with the posterior end of the pharynx. And so the model that we came up with to try to explain how this muscular tube could actually produce a cavitation bubble was premised on this experience that many of you may have had with trying to suck a very viscous liquid through a limp straw. So if you've ever tried to drink booster juice or a smoothie through a paper straw that's starting to get limp, if the tip of the straw starts to close the least little bit and you suck harder, it just pulls it closed even tighter. So what we think is happening here is we think of this muscular tube as kind of a straw that's limp. And initially, so this is just a schematic of the series of steps, somehow, possibly due to circular muscle contraction or increased hydrostatic pressure inside these muscles, the opposing sides of the pharynx here are pushed together. So in cross section, this pharynx is split, so it can actually come together flat, but it's very elongate in one direction. So these sides are pushed together, and then once they're pushed together, the muscles in the posterior part of the pharynx can start to contract, and that does two things. It closes the tip of the limp straw even more tightly, and then these muscles can continue to contract and store energy. And this is, you can almost see these worms tense up before they strike. And then the strike, we think, happens extremely fast when muscles in this very distinctive region of the pharynx, very dense radial muscles, pull apart the edges of the pharynx, the collapsed tip of the limp straw, and they pull apart so fast that the water cannot make it into that space as fast as the muscles pull apart, creating a cavitation bubble. And then as the water rushes into that vacated space, it eventually collapses against the another surface of water on the inside. And then the pharynx eventually fills up with water as these radial muscles continue to contract. So these animals are not so easy to collect. We're hoping to get some more in a future dredge and get them, try to film them under a camera where we can try to actually visualize the production of this cavitation bubble, or maybe even film them in the dark in the hope that we see a flash of light associated with snapping to confirm its cavitation. But anyhow, that's the model that we have for how how a simple muscular tube can create this kind of rapid movement and snapping sounds. So this leaves us with the Bamfield mystery. So after getting back from a summer of hunting snapping shrimps all over the world, we threw a hydrophone in the water at Bamfield and heard this raucous sound. And then last summer, I made an attempt to try to find some of these animals. This sound, for any of you that have ever been diving in the tropics, sounds very similar to the popcorn sound you hear due to snapping shrimp that are widespread in tropical waters, but there are no snapping shrimp in Barkley Sound, so we don't know what this creature is. So you would think it would be fairly simple to just go out and find a creature that's so abundant and making such loud sounds, but it's much trickier than you might imagine, because unless you hear the sound, you don't know if that's the animal. So I tried to do some sampling this summer out in this area where popping sounds were really loud and frequent. And the first tide series, I went out and collected some stones with fairly complicated and diverse fowling communities and just started digging through them, looking for creatures that might be in the right size range to make a snap. And there was nothing that looked really compelling. There's a scale worm that has this really weird behavior of sticking a medial tentacle in the middle of its mouth. Another worm that was big enough to snack, but we doubt is likely to do so. And I was unsuccessful that whole first tide series. The second tide series, I went out, same thing, collected a rock with fowling community. And this time I tried to be smarter by subdividing the rock fowling community into different buckets. So sea squirts in one bucket, algae in another bucket, some of these encrusting tube worms in another bucket. And then I was trying to figure out which bucket had the snap. And at the end of the first day, I didn't notice any snaps. And I was about to discard this rock because I had removed almost everything from it. But as I picked up that bucket and started to carry it down to the shore, and I jerked it, there was a really loud snap. And I was so excited because I now knew I had a snapping creature in a bucket with almost nothing else, just this rock. I went back the next day and I spent like 6 hours looking closely at some of these other things, not the other subsamples, not finding anything, taking this rock out, removing about a third of what is left, putting it back in the bucket, not finding anything, giving the bucket a jerk, still confirming that the bucket snapped. And then after about 4 hours of doing this, there's nothing left on the rock. And either it's a creature that bores into the rock, which would be spectacular, or this awful voice in the back of my head said, I wonder if it could be the bucket. And so I took the rock out of the bucket and gave the bucket a little jerk, and it snapped. And so for 2 tide series, probably 10 days of my life this summer, and you can see this plastic bucket, it's a plastic handle in a plastic bucket that makes those snaps, not the metal handles in the black buckets. For 2 weeks, I was chasing a phantom snap. So just let that be a lesson to you. So anyhow, the hunt goes on. I don't know what this creature is, and I'll be out there in a couple of weeks. Maybe if I get some time, I'll do some more hunting. So what I'd like to do to finish up, I still got a few minutes here. When Dave invited me to give this talk, I started to think about maybe trying to summarize a 40-year research career at the U of A, and I was really struggling with how to try to do that in a way that wouldn't be just stupefyingly boring. And so I thought, well, let's try to do it in just one slide. So what I've done here is tried to summarize a 40-year research career at U of A in one slide. And I think there's three useful lessons that come out of that came out of this exercise, even for me. The first is, as you look across all of these topics, these are sort of 6 major areas where I did work over the years. In the early years on snail shell form, genetics and plasticity. These are 2 variants of a single species of snail that change form depending on the condition in which they're grown. The fluctuating asymmetry that Dave mentioned, which is statistical variation about perfect symmetry thought to arise due to developmental instability. That occupied almost 10 years of my life. And then after that, that was followed by an ongoing interest in really conspicuous left-right asymmetry because, and I'll spare you the details, but I think there's evidence looking at phylogenetic patterns that very strong evidence for a genes as followers mode of evolution, sometimes called genetic assimilation. Then at sort of the end of my fluctuating asymmetry career, I sort of stumbled on this phenomenon of selective reporting that Dave also mentioned. And then finally, the last 15 years or so, 20 years, mostly I've been interested in barnacle appendage plasticity, both legs and penises, and also barnacle reproduction, some of which actually Dave was involved with. And then finally, in the comparative morphology of extreme movements. So one of the first things that leapt out at me looking at all of these is that among the top 10 public most highly cited papers of my career, there were 1 or more from each of these 6 different topics. So if you look at this overall pattern, it looks far more like a random walk. There's not much that connects these different types of, these different topics together. Plasticity, fluctuating asymmetry, conspicuous asymmetry, selective reporting, and so on. But in each one of them, you know, I found something that I found to be really quite fascinating, and that ended up being a paper that received, that ended up being quite highly cited. But you might wonder how it is that I came to find these other subjects as being interesting. And that, I think, might be the most valuable lesson of all from this. I first got interested in snail shell plasticity by chance. I just put some of these individuals of this species in a tank with food and fed them for a few months, and discovered remarkably that over a month or 2, they started to change shell form in the lab. That eventually led to a discovery that snails could smell crabs and produce more well-defended shells in response. The fluctuating asymmetry that resulted in my single most highly cited paper, I got introduced to by a random encounter with a graduate student when I was giving a seminar at Johns Hopkins University. Just sat me down. He wanted to tell me about his thesis work on fluctuating and asymmetry. And I thought, shit, that's really interesting. And I just started reading about it. And then eventually, with Kurt Strobeck, we discovered this way to do the analyses in such a way that made them a lot more robust. But that got started with a random encounter with a graduate student while giving a seminar. The interest in conspicuous left-right asymmetries just came about from pursuing a vague curiosity about patterns of asymmetry variation, where in some species you have symmetrical animals, in others you have some individuals are right-sided and some are left, and others they're all fixed in one direction. And I was very curious about the relationships among those. And eventually that led me to realize that one could make a case that that this mode of evolution, genes as followers of genetic assimilation, is actually surprisingly common. But that came about just because of this kind of vague curiosity about patterns of variation in left-right asymmetry. How did I end up involved in studying selective reporting and publication bias? I simply read a paper of Anders Moller. Moller and Thornhill did one of the first meta-analyses of fluctuating asymmetry data. Within a week or two of that being published, I whacked the results all into a spreadsheet, just saying there must be something interesting here. And you do a simple graph of effect size versus sample size. All of a sudden you realize there's a graphical way to detect selective reporting. So it was really this, again, this vague curiosity was what pattern might emerge by looking at the data from this meta-analysis that led to a pretty, I think, pretty significant discovery about selective reporting and publication bias. This very long period of time working on barnacle leg plasticity and barnacle reproduction was catalyzed by a casual conversation with a student at Friday Harbor Labs. He said, you know, have you ever looked at barnacle legs? They really vary a lot between exposed shores and protected shores. So again, just this random encounter. And then finally, this last area, which I talked to you about today, is really almost entirely a result of a past postdoc of mine, Tomonari Kaji, who's this exquisite comparative morphologist. And he's the one that really got me interested in the mechanics of these super fast movements. So what's the sort of the takeaway from this that maybe some of you might find helpful? And this is, I've done this enough times that I would trust it, which is trust your gut. When you stumble on something that seems interesting, even if you can't initially say why it's interesting, spend a little bit of time reading and thinking about. You might be really surprised by what you find. I also found it's incredibly valuable to encourage your students to do that same thing, as well as to listen to what excites them. So they're trusting their gut, they're being excited. Listen to what they have to say. And then for NSERC proposals, there's always this battle between methods and questions. And I would have to say that in my experience, both in many grant proposals and sitting on the NSERC committee, and Dave, maybe you can comment on this as well, but I think cool biology trumps sophisticated methods every time. So make sure that in NSERC proposals that you emphasize what is really cool about what you're doing. And then finally, I guess I really should finish by saying how incredibly grateful I am to have worked at this amazing university, which also happens to be in a civilized city and a civilized country. So I think a lot of my success has been because of the conditions that U of A has created here for me. So with that, I have a few people to thank about this work. My postdocs, Tomonari Kaji and Arthur Anker, both worked on the Snapping Shrimp project. Christian Wertner from Germany, a couple of Japanese colleagues, Ryotaro Goto and Isao Hirabayashi, who introduced me to this fabulous snapping worm system. And then again, Tomonari Kaji, his friend in Japan, Keiki Koichi. And then finally, the members of the Patek lab at Duke, who did a lot of the kinematics of amphipod claws. And with that, Dave, I'm happy to turn the hosting over to you to take questions from the audience. Thanks very much.
Well, I just have to say, Rich, that was fantastic. That really was. The last 5 minutes, wonderful, wonderful. Yeah, and one of the one of the negative consequences of doing this by Zoom is you can't hear everybody applauding, but I think we're all applauding inside because that really was both a fascinating story about some natural history and some biology, but the perspectives you've just shared with us are that I'm really touched by that. So with that, I'll turn things over to questions. We've got a few minutes for some questions, and I'm sure there must be some because that was really cool stuff. And I would encourage people either to use your blue hand, and I see Martina first, and yeah, we'll try to do it that way. Martina.
Thanks, Rich. That was the absolute highlight of my day. Truly, it was just great. And you know, I was actually stunned by what you said about the hydrophone recording, and that you really didn't know what was making that sound. And so I guess I mean, it's stunning, but it's not surprising that, you know, you put a hydrophone somewhere and you hear these sounds that you don't automatically know what they are. And I think our whole world is full of that when it comes to sounds. But so what do you actually think it is? And because it's also a familiar sound, right? Like you hear that all the time. So what do you think it is?
You know, after we discovered this worm that could snap, it basically removed all constraints on what creatures might be able to do this. So I don't know. I mean, my hunch is it's probably a crustacean of some kind, just because the claws are sort of pre-adapted for eventually being able to jet water. I would dearly love it to be true of this group of crustaceans called taneids, which are even smaller than amphipods, but many of them have these paired claws that are quite large relative to the size of the body. And so I would love it if we found one of those that was the creature that was able to do that. But beyond that, I can't say. We've basically ruled out fish because the habitat where the hydrophone, where the intensity of the snapping is loudest and most frequent, there aren't any fish or any obvious fish. So that pretty much means it's an invertebrate of some kind. But honestly, beyond that, I don't know. And that's why the hunt is so much fun. But it also is why the hunt is so bloody hard, because it could be anything. So initially, I was trying to search for things in a certain size range that seemed like they might be big enough to do it, but nothing really promising yet. But you should come out to Bamfield sometime.
I will. I was talking to Sean this morning about my next, when I can get out there. So anyway, this was great. Thank you so much.
Well, nice to meet you. I'm sorry we haven't met before.
Likewise, Rich, likewise.
Very good. Thanks, Martina. I see Declan is next, the next blue hand I saw. Stephanie, I see yours.
Hi, Rich.
Hey, Declan.
That was fantastic. How are you doing?
Good, thanks.
I was wondering, I mean, really interesting, and you talked about the latching mechanism, the ridge, etc. But do you know if there actually are any differences in the muscles themselves in those organisms? So are there any differences in the isoforms for things like actin, myosin, troponin compared with other organisms?
I don't know, but that would be an absolutely fascinating thing to look at, particularly in the snapping, the shrimp that have snapping slip joints, because there a portion of the closer muscle is involved in energy storage, and then it's only a small part that's involved in causing it to shift to release all that stored energy. It's possible that someone's looked at that, but I don't know, and if they haven't, it certainly would be something worth doing.
Yeah. Okay, great. Thank you.
I mean, for example, they might be slow contracting muscle, for example, because there's no movement really. It's just stored energy. And then the movement all comes from the slipping once it slides over the ridge.
Right. Yeah, that's great. Thanks.
Okay, I think I had Heather's blue hand, and then following that, I know Stephanie is indicated she has a question. So Heather.
Sorry, chat. My microphone is horrible, so it's in the chat.
Oh, got it. Sorry. Okay, I'll read that out loud. Sorry, crappy microphone, so I have to type. Given the power of those snaps, did you ever see damaged claws on male amphipods or snapping worms with their pharynxes blown out? Finally, are there such things as directional hydrophones?
Um, so that's a couple of questions there. Um, no, we never saw, even in snapping shipboard amphipods that I ever see, at least in, the few hundred that I've looked at, have ever seen a damaged claw. And I think that's because it's not impact that makes the sound. It's the jetting water in the cavitation bubble that makes the sound. And that sound is being produced at a little distance away from the tip of the claw. And I don't know about the worms. I haven't actually never seen one of these worms alive myself. It's only my Japanese colleagues that have seen them, and they've only seen maybe half a dozen. So there's too small a sample size there to say. And I don't know. I don't know. Maybe Martina could answer this. I don't know about directional hydrophones. What I have learned over the years of doing this is that underwater sound is freakishly complicated because there's so many echoes. And I mean, the hydrophone is near the boat. Well, their sounds are going to echo off the boat. Heaven forbid you put a hydrophone in an aquarium with something that snaps, because you get all these echoes just from the container. So I don't know about directional hydrophones. Google might, but yeah, that would be really helpful, because then you could narrow down your search to areas of interest.
Okay, thanks. Steph, you did stick your physical hand up.
Hi, Rich. It was a
Hey,
pleasure.
Jeff.
Hello, to hear your seminar and finally to hear you talk about your research. That was wonderful. And
Thanks.
I thoroughly enjoyed the last few minutes as well, to see the diversity of things you've done. It gives one who's maybe at the beginning part of that trajectory some hope that there'll be a lot of diversity of things to come. But I wanted to follow up and ask about the soundscape part, because
Yes.
I wondered if you had an idea of the spatial or temporal variation in that signature, and whether that might tell you something about the communities that might be driving it. Like, do you see any differences with night and day, or have you tried different habitat types to
So
see if
this is for that amphipod in Banfield, you mean?
Mhm.
Yeah. So I have done part of that. I had one consequence of COVID last summer was that the summer student that was going to help me find these animals couldn't come to Banfield. But fortunately, she was super good programmer, and she was very, very familiar with R. And so I hired her as a summer assistant, and she worked for 2.5 months at home developing an R script, which is just this breathlessly cool script. So you put a hydrophone in the water, you hit go, you record 2 minutes, you go to another site, record 2 minutes. And I went to maybe 15 or 20 sites in Grappler Inlet, actually. And she wrote the script where I just put the sound files into a folder. I specify a couple of things to do with spacing of pops, and then I say hit go away and come back 6 hours later, and I get frequency distributions of the number and intensity of pops for those 2-minute recordings for all 12 or 15 sites that I went to. So that's a long answer to your question, which is yes, it varies quite a bit spatially in Grappler Inlet. In the narrows there, right between Port Desire and the head of Grappler Inlet, it's super loud. That's where I was actually sampling. And even right in the mouth of Grappler Inlet across from the director's house, it's fairly loud. It also happens to be pretty loud right under the dock at the marine station there. So probably maybe this fall while I'm out there, if I get some time in between online lecturing and online labs, I'll see if I can't get some samples off the dock. In terms of day-night, I haven't done that yet. In terms of season, I haven't done that yet. When I go back out there in November, I may try again to see if there's any difference there compared to summer. But this script that she's written at least allows me to do it now in quite a quantitative way.
That's incredible. It would be interesting to see, I mean, if you did a spatial analysis mapping on some other maybe drivers across those sites to
Maybe.
look at
Yeah.
correlates. So very cool. Thanks.
And just as an aside, that student who was so wonderful, I met as a student in the fall program last year. So I've told you about the value of teaching out there and using it as a way to recruit really good students, and that was an incredible success story for me.
Great. Very cool.
Great. I have one more I see here in the box from Jan Muri.
Yes.
Did you see that?
Yes, I did. Jan. Hey, Jan.
I'll just read it for the record. So is all the noise one hears when snorkeling on coral reefs due to snapping shrimp rather than fish scraping coral, which I had been told at some point.
I suspect that the vast majority of the sounds that you hear snorkeling or scuba diving on coral reefs are probably 80%, 90% snapping shrimp, and maybe 10% stomatopods. There's some hammering stomatopods that end up creating cavitation bubbles when they hammer. But yeah, I would say the vast majority of those popping sounds are from snapping shrimp in tropical reefs. The scraping noise is a totally different sound. It's more, if you record it with a hydrophone, it's more like a rumble than a snap. There are some, actually Steph could probably answer, there's some parrotfish that I think bite the tip off a cropora, and maybe that makes some snaps, but I wouldn't be nearly as many as snapping shrimp.
Yeah, well, it certainly, the noises, as I recall, certainly sound like this very repetitive snaps that the shrimp are making.
Yeah, no, it's one of the first, I still remember my first time snorkeling in high school. I couldn't believe it when I put my head in the water in the Florida Keys. It's just like you stick your head into a popcorn popper.
Yeah. Thanks.
Somebody's just pointed out there's another question I missed in the chat from Lean. What might be the function of the snap in the worms?
Oh,
that's actually something that occurred to me.
Yeah, bless you, Lean. So we don't know for worms or for amphipods. It's not clear that the snap is functional. It may just be a byproduct of super fast movements. In snapping shrimp, though, different species use them in different ways, and there's actually not that much known. They are certainly aggressive, so they fight with each other, because if they can actually damage an opponent, if that cavitation bubble is close enough to them, so they use them in aggressive interactions or competitive interactions. There are some amazing clips on YouTube of them stunning shrimp and possibly stunning other prey, so they can use them feeding. You know, I don't know if this has actually been demonstrated. Many people suspect that they signal to one another, so they sort of define their territories by signaling using these sounds, but I don't know that that's ever been quantified. So there, in snapping shrimp at least, where it's super well developed, and there's also several hundred species in which snapping occurs, it's actually used in quite a variety of different ways.
Very good. Okay, Rich, I think that's a wrap.
Okay.
I want to thank you so much for reminding us of something I think that's really important, which is many of us are here because the curiosity and wonder about the natural world turned us on to biology when we were students.
Correct.
And we were inspired to do something, to discover something when we were graduate students. And that brought us where we got to today as academics. And I think sometimes when you're dealing with all kinds of things that I deal with on a daily basis, we forget about the curiosity and wonder, which is why we're here and why so many of our students are here. So I want to thank you for reminding us of that, and for reminding us of the value of that across your career, and keeping your mind open to opportunities and to questions that might arise. And I think you made a really good point about NSERC, and at least the culture. When I was a co-chair at the Ecology and Evolution group, it was very much, as you said, it's about good questions and interesting biology, and methods do come second. Yeah, and I fully.
I mean, it has to be plausible. It has to be doable, but it's not the sophistication of the techniques. It's the significance of the questions that matter more.
Yeah, yeah, and I think your track record is a testament to how what the impact of that can be. Yeah. So I did want to ask you what the industrial applications of your snapping claws were, but maybe I'll leave it.
It's not clear. Most engineers don't like cavitation bubbles because they destroy things.
Right, or they expose your submarine to somebody else, right?
But I mean, there is a potential engineering application because this whole notion of a torque reversal joint where you can store energy and then close it much faster than you could, let's say using a servo motor or something like that, that could potentially be useful, but I'll leave it to the engineers to come up with an application.
Okay, thank you, Rich. And with that, I just want to remind everyone that there's one more chairs lecture this semester. So December 10th, Keith Tierney will be our last speaker of 2020. And finally, Rich, you will receive a coveted bottle.
Whoa.
Yes, that's right. A Department of Biological Sciences inspired discovery for a better world, which you've just done for us. Bottle, and you'll receive it in your mailbox or something. Linda will figure that out. But
all right. Thank you so much.
And with that, I want to say thank you. Good to see you, everyone. And
good to see everybody.
Yeah, tip my hat to you, Rich. Thanks so much for doing this.
Okay. Well, thanks for your attention.
Bye, everybody.
Bye-bye.